The American Brain Tumor Association is pleased to welcome you back to our webinar series. Our webinar today will discuss immunotherapy for pediatric brain tumors. My name is Antoinette Tu, Program Manager here at the American Brain Tumor Association, and I'm delighted to introduce our speaker today, Dr. Elias Sayur. Dr. Sayur is a board-certified pediatrician and oncologist who has taken care of numerous patients with aggressive malignancies and is principal investigator of the RNA engineering lab at the University of Florida Brain Tumor Immunotherapy Program. Dr. Sayur is an NIH-funded investigator who has received numerous awards for his work with RNA nanoparticle vaccines, including the ABTA Discovery Award. Thank you for joining us, Dr. Sayur. You may now begin your presentation.
Thank you. Thank you for inviting me to speak here today. Again, my name is Elias. I'm going to be talking today about immunotherapy for pediatric brain tumors. I spend 80% of my time doing research, looking for novel ways to harness the immune system against cancer, pediatric brain cancer specifically. 20% of my time I spend in the hospital taking care of patients. And it's really my experiences as a pediatric oncologist that have led me down this research path. And this is epitomized by malignant brain tumors in children. Malignant brain tumors in children are often very refractory now. Malignant brain tumors has actually overtaken leukemia as the most common cause of solid cancer death in kids and most common cause of cancer death in children. And really, even in cases where we have good outcomes, these therapies have side effects that are really inappropriate. Children with developing brains that receive radiation therapy, 36 gray of radiation to the cerebrospinal axis in patients with medulloblastoma, which can really affect the developing brain, combination chemotherapy, these things that can affect your hearing, aggressive surgeries in the posterior fossa at the back of the brain for patients with tumors back there can affect one's ability to walk or talk. And so even when we attain a cure in patients with malignant brain tumors, unfortunately, it can come at a big price in that many patients may lose their ability to walk, talk, hear sometimes, and certainly the risks of radiation. So the need for new therapies is paramount. And as a pediatrician, I've always taken to the idea of vaccines and the immune system. And certainly, immunotherapy has really come to the forefront recently in the last few years as a very promising modality that can be harnessed to mediate substantial regression of tumors in many adult cancers. And really, what we're trying to do here, and many people around the country are trying to do, is harness some of these strategies for children with brain tumors and for other childhood cancers. So I will discuss some of the tools that are being used in the context of immunotherapy for pediatric brain tumors. I have no stock patent rights or employment with any company, but I do have some preclinical laboratory support from Immunotherapy that I want to disclose. So cancer immunotherapy was Science Magazine's Breakthrough of the Year in 2013. And this was really an amazing accomplishment for the field of cancer immunotherapy because it really encompasses the entire field of science. To give you an idea, the year before the Science Breakthrough of the Year was the so-called God particle, the Higgs boson that was discovered in physics. The year after, the Breakthrough of the Year was landing a probe on a comet. So the idea that cancer immunotherapy was recognized in the entire field of science was quite remarkable and really showed a lot of the progress we've made over the years in developing this idea from something that has achieved tangible benefits in patients. And when we look at the history of cancer immunotherapy, it's not brand new. People have been studying cancer immunotherapy for well over 100 years now. And it really started in 1863 by the experiments by a man named William Coley, who's really considered the father of cancer immunotherapy. And what he did would probably be considered unethical today. But what he did was he took bacterial products and he would inject them into tumors. And so I don't imagine there was, he got approval. I don't know if he got consent for this. But what he noticed when he would inject these bacterial products into patients' tumors, he would notice that the tumors would shrink away. They would start to dissolve and remit. And he actually noticed that not only the tumors he would inject would improve, but he noticed that patients that had distal metastatic disease, that those metastatic tumors would also improve from just the injection of this bacterial products at the local site. And so what he was witnessing was really the first evidence that the immune system can be harnessed against cancer. And what was happening is these bacterial products were being injected in the tumor, and they led to an immune response against first the bacteria, but an immune response that also then spread to the tumor itself, allowed tumor killing. And by getting tumor killing, this allowed the immune system to recognize the contents of the tumor as foreign. And in some cases, the immune system attacked other sites of the tumor, such as these metastatic sites. So this was the first evidence that the immune system really could be harnessed against cancer. But bacterial products has a lot of concern, and they're certainly can lead to infections and other risks, and this was certainly not a FDA-approved therapy. And people have tried, though, to reproduce these results in other contexts. And I'm not going to go through the entire history here, but basically there have been seminal discoveries over the course of the last 100 years that have really led us to 2010. And in 2010, what happened is we had the first FDA-approved drugs for cancer. This would be sipuleucel T for castrate-resistant prostate cancer, and this would be ipilimumab, the checkpoint inhibitor that has shown pretty dramatic effects in patients with metastatic melanoma. And so we'll go over some of these strategies, what they mean as the talk continues. But I've always wondered if the immune system can be used, harnessed against cancer, and certainly we have drugs that can actually unlock the immune system now against cancer. I've always asked, why doesn't the immune system do this naturally? Why doesn't the immune system just get rid of cancer, before it happens? And a lot of people believe in this idea of cancer immunoediting, meaning that this actively happens, that the immune system is constantly getting rid of cancers in all of us. But then what happens in patients where there is cancer? So it would seem that the immune system does actually develop a response against cancer. And this would be called the elimination phase, where a cancer may be starting and your immune system recognizes it as foreign and develops a response against it. Unfortunately, what happens is as you kill these cancer cells, new ones sprout up. This is evolution in real time. New cancer cells sprout up that have resistance mechanisms against your own immune system. And you have this equilibrium phase where these cells are evading the immune system, but the immune system is killing the cells that are very sensitive against it, that don't have those resistance mechanisms. But then over time, the immune system ultimately loses because more and more of the resistant cancer cells are forming. And this is the escape phase, where cancer is actually escaping the immune system. And not only is it escaping the immune system, what happens in a lot of solid cancers like brain cancers, glioblastoma, what happens is these cancer cells can actually manipulate, contort the immune system to work for it as opposed to against it. So these cancer cells can now recruit regulatory immune cells that turn off inflammation, that turn off the killing capacity of other immune cells that would recognize it as foreign. And so in advanced cancer, not only is the immune system not able to fight off the cancer, it actually is working in tandem with the cancer to potentiate the growth of that cancer. And this is part of this concept called cancer immunoediting. But when we talk about the immune system, it's important to recognize that the immune system has a lot of diverse roles and importantly, it has different arms. And so broadly speaking, we can really divide the immune system into these two arms called the innate arm and the adaptive immune arm. And so the innate arm, this is your first line of defense. So anyone who develops any infection, you can develop fever in the first couple of days of an infection, you feel sick, but the first line of defense are these cells that I've shown here: macrophages, dendritic cells, natural killer cells. These cells are very important at finding the invader and then presenting that invader to the adaptive immune system. The adaptive immune system can now make a very, very specific response against that infection. And the adaptive immune response, broadly speaking, can really be divided into T cells and B cells. B cells make antibodies that can get rid of things like bacteria, and T cells make killer cells that are very, very good at finding out what pretends to be you when it's not. And viruses do this all the time. Viruses infect our cells, and they pretend they're us when they're not. They hijack our cells. Our cells make viral genes. They don't make our genes anymore. And the viruses just live in our cells and take them over. And it's the T cells. The T cells are very, very important at recognizing the cells that are foreign, those cells of ours that have been infected by virus, and get rid of them. That's what the T cells do. In many ways, I think of cancer as a very similar entity. Cancer pretends it's us when it's not, just like a virus. And T cells can actually be programmed to recognize cancer as such and get rid of it, in the same way T cells can get rid of viruses. And T cells probably naturally do this in a lot of patients, get rid of cancer before it starts. But unfortunately, with cancer immunoediting, they fail in advanced cancers and ultimately become subverted to the immune system, that is, to enable the cancer. And when we talk about T cells, if T cells can do this, if T cells can get rid of viruses, they can get rid of cancer, how can we activate them? And so naturally, T cells are activated by a cell called an APC, which stands for antigen-presenting cell. That APC has to present the parts of the tumor or the virus to the T cell shown in purple here. And that T cell, for it to become active against those pieces of tumor or virus, there needs to be two signals. The first signal comes in the form of the MHC-presented tumor or viral piece, that what we call an epitope. And that has to be presented to the receptor on the T cell. But there needs to be a second signal, what we call a co-stimulatory signal, between the APC and the T cell. And those two signals together can turn that T cell on. The reason there needs to be two signals is this is nature's way of regulating T cells so that we don't have autoimmune disease. So these two signals are very important to turn that cell on, and when that cell turns on, the entire lifespan of that cell will be to find that piece of tumor or virus or whatever it's been activated against and kill it. These T cells will divide. They will make something called cytokines, which allows more immune cells to come into the fray, and they will proliferate, and they will find the tumor and get rid of it. And so T cells are really important in the context of cancer immunotherapy, and they're really important in the context of malignant brain tumors. And the reason they're so important is because T cells, activated T cells, can cross the blood-brain barrier. This is a huge problem in the context of cancer therapy in patients with malignant brain tumors. How do you get therapies across the blood-brain barrier? Traditionally, it's chemotherapy, drugs. These drugs, unfortunately, we have to give more and more of the drug to get some penetration across the brain tumor. You know, so when we talk about things like cisplatin and temozolomide, they can penetrate, but not very effectively. And so what we end up having to do is give more and more of the drug so we can get some effects, but unfortunately we can get a lot of side effects. With the immune system, these T cells can naturally cross the blood-brain barrier when they're activated. They'll find the tumor antigens, they'll kill them. But unfortunately, cancer is very, when cancer is advanced, as it is in many brain tumors like medulloblastoma, glioblastoma, unfortunately, these tumor cells have really transformed the landscape of the immune system. They transform the landscape of the immune system to something that's very regulatory. And so for these tumor cells, like glioma cells, to be recognized by the immune system, they have to present something called MHC class molecules, pieces of it that the immune system can recognize as foreign. And what these cancer cells do, they're very smart, is they get rid of this. They get rid of this MHC class presented molecule. And they also secrete cytokine signals that can decrease the activation or the killing capacity of these T cells, both the CD4 and CD8 T cells, which are both very important for T cell responses. And so how do we fight this? If cancer is so good at manipulating the immune system, how can we fight this? And as much as I hate to say it, chemotherapy has been used traditionally in the context of immunotherapies to do just that. Chemotherapy can actually get rid of a lot of these regulatory immune cells. The immune cells that the cancer has manipulated to work in its favor. The chemotherapy gets rid of a lot of those cells, and the damage it does to the tumor allows new immune cells to pick up those dying tumor cells and develop immune responses against them. And so chemotherapy can be used in conjunction with immunotherapy to potentiate the immune response that's desired in patients. And I hope there will be an era where we're not using any chemotherapy, but right now many of these trials embed chemotherapy with immunotherapy exactly for this reason. And so this brings me to this whole cycle of cancer immunity and immunotherapy that we're all trying to do to redirect this, in adults, in children, for patients with malignant brain tumors. What we're trying to do is develop this self-sustaining cycle of cancer immunotherapy. And what we're hoping to do is, by redirecting the immune system, what we're hoping to do is, by just getting the process started, we're hoping that the immune system can cause T cells to recognize and kill tumor antigens. And when that happens, the endogenous, the natural immune system inside the patient, we're hoping to reboot that, kickstart that. Because once we can get the process started, if we can get T cells to recognize and kill tumor cells, then the natural immune system, things like dendritic cells, can come in. A patient's natural immune cells can come and pick up those dying pieces, go to the lymph nodes, present to other T cells, the patient's own natural T cells. And those T cells can come in, kill tumor, shed antigens, and this cycle can continue again and again and again for what we hope would be complete tumor regression. But the issue is, in cancer, this is not kick-started. In fact, the immune system is working with cancer. So we have to kick-start this to get this cycle going. And how do we do that? We can do that with vaccines. We can introduce a dendritic cell vaccine or a peptide vaccine that can present pieces of the tumor to T cells in the lymph node and get this process kick-started. We can make T cells. We can take T cells from the patient, engineer those T cells outside the patient's body, and then give it back to the patient. T cells that recognize the tumor here, which then can shed these antigens and then get this process kick-started. Another way is by blocking the immunoregulatory elements. Cancers have very, very sophisticated means of turning off the immune system. And we've shown if we can just block those regulatory mechanisms in cancer, if we can just block those regulatory mechanisms, we can actually allow the patient's natural T cells, we can actually unlock their potential against the patient's cancer and get this process kick-started. And so I'm going to talk about each of these areas for pediatrics in this talk. And so when we talk about pediatric cancer immunotherapy, much of the landscape has been divided in these areas. Adoptive cell therapy, again, which is making these T cells, either genetically engineering them or activating them outside the patient's body, giving them back to the patient. We can also talk about cancer vaccines, taking pieces of the cancer, putting it inside an immune cell or in the form of a vaccine, giving it to the patient to generate a T-cell response inside the patient. Also, people are using oncolytic viruses, so viruses that can be injected straight into the tumor that can lead to an immune response more globally against the tumor. And unlike Dr. Coley back in 1898 experiments where he gave bacterial products, these are viruses that are what we call attenuated, meaning they're not the full virus. They're viruses that have been gutted from their ability to cause harm to healthy cells, but have the ability to infect cancer cells. And then lastly, we'll talk about targeting immunoregulation. These things like immune checkpoints that once blocked can unlock a person's natural T cells, their natural immune responses against their underlying cancer. So adoptive cell therapy, we'll start here. Much of the work in adoptive cell therapy has come from the work done by Dr. Steven Rosenberg at the National Cancer Institute. If Dr. William Cooley, the father of cancer immunotherapy, Dr. Rosenberg is one of the modern day fathers of cancer immunotherapy. He's done these experiments in patients with melanoma where he's taken out the tumor, as much of the tumor that he can access. And he's basically taken out the T cells from that tumor and grown them up with cytokine signals that allow those T cells to become very angry. And then he's given back those T cells to patients. So basically trying to take the T cells that are naturally inside a patient's tumor and redirect them, re-energize them before giving them back to patients. And he's shown dramatic responses in patients with melanoma. Very, very impressive regression shown here. Here's another patient with a very dramatic regression of their metastatic melanoma treated with this type of a therapy. And so what they're doing again is when they access this tumor, they fragment the tumor mass and they're able to grow up the T cells, these T cells, in a very outside the patient's body in culture conditions that allow these T cells to grow and become robust and very active against a patient's underlying melanoma. And then these T cells are given back to the patient in the context of chemotherapy to get rid of the regulatory elements and allow these cells, these angry T cells, to go to work. And in so doing, this has led to some very impressive regressions that I've shown. However, in brain cancer, this is the challenge. Unlike melanoma, which is very easy to access and get these T cells out of patients because it's a skin cancer, brain tumors are much more difficult to access. We can't get nearly the amounts of T cells out of a brain tumor, especially if in these small biopsies that we can in melanoma. So how can we use adoptive cell therapy in patients with brain tumors when we have very limited amounts of tissue that we're working with? So one strategy is actually to take T cells from patients, that is not T cells from the tumor, T cells from the blood of patients, to collect T cells from the blood of these patients and actually genetically engineer them to fight cancer. And so in these patients from something called the leukapheresis, we can extract white blood cells and take those patients' T cells and actually genetically engineer those T cells using a virus that reprograms that T cell to fight a patient's cancer. And what we do is after reprogramming these T cells, we can give them back to patients and allow those T cells now to fight that cancer. And this is really what's called these CAR T cells. These genetically engineered T cells are called the CAR-modified T cells. The term CAR stands for chimeric antigen receptor. This is a genetically modified T cell, not normally found in nature, as far as I know. And what people are doing is they're essentially making these T cells super active and bypassing the way you would normally activate a T cell. So remember, to activate a T cell, you need an APC that presents these two signals to a T cell to get it to activate. Again, this is nature's way of controlling the immune system so that it's a very specific response. But what these CAR T cells do is they bypass this completely. What these CARs do is you're taking a signal that would naturally recognize a cancer cell, and you're fusing it to the activating domains of the T cell. So when these cells are given back to patients, the minute this surface sees the cancer cells or the cancer pieces, the cancer antigen, this T cell will recognize that cell as foreign and kill it. There are other ways, though, of activating T cells against a patient's cancer. This is something we do here at the University of Florida. So from as few as 500 tumor cells from a biopsy, what we can do is we can extract RNA. So RNA is a messenger protein, it's a messenger nucleic acid that is made from DNA. So, DNA has all the information inside these tumor cells. And that DNA needs to be made into something called RNA before it can be made into a protein. And we can extract that information, that RNA, from the tumor cells. And so this is the piece of information inside these cancer cells, the genetic information that's actually being expressed inside these tumor cells. And what we can do is we can take that RNA and put it inside these dendritic cells. Dendritic cells are really the master cells of the immune system. They're APCs, antigen-presenting cells, that can really activate T cells to do the job they need to do. And so what we can do is from this RNA educate these dendritic cells that we can obtain from patients. So we can take dendritic cells from patients, we can put this tumor RNA inside these dendritic cells, culture them outside the patient's body with the patient's T cells. And then we can give both the T cells, these T cells that are now activated, they've been activated not by being genetically engineered, they've been activated by being presented to by these APCs, these DCs, in culture conditions. So this is what naturally happens inside the patient's body, and we're just taking it and doing it outside the patient's body. Essentially taking dendritic cells that have tumor information and culturing them with the T cells that can now become active against a patient's individual cancer. And now we can give these T cells back to the patient with the dendritic cell vaccine, so they're getting to both a vaccine and a T cell product. And we give this in the context of stem cells with chemotherapy, and then we look for patient survival outcomes. And so these T cell strategies are some of the most common ways of utilizing adoptive cell therapy against patients with pediatric brain cancer. In terms of studies, there's a phase 1 study looking at HER2-specific CAR, so these are the genetically modified T cells, in patients with glioblastoma. And while the study right now is only for patients more than 18 years of age, it's being done at Baylor Texas Children's Hospital. The principal investigator, Dr. Nabil Ahmed, is a pediatric oncologist, and this very well may be in pediatric studies in the very near future. And then here at the University of Florida, we're looking at a phase 2 study in patients with recurrent medulloblastoma, where we give them those RNA-loaded dendritic cells with the T cells that I just described for patients with recurrent and relapsed disease. So shifting gears, I want to talk about cancer vaccines. One of the issues with adoptive cell therapy is adoptive cell therapy requires a lot of a lot of engineering outside the patient's body. That engineering can be very costly and very complex. And for many patients, it may take a long time before they get their product. So the advantage of what we do with the dendritic cells and the T cells is it's a very personalized therapy against a patient-specific tumor, but it can take months to generate. What would be much more attractive is having a therapy that's available in real time for patients, a vaccination approach that isn't as costly or complex as some of these cell therapy measures that I just discussed. And there is such a thing as passive cancer vaccines. And so in several different types of cancers, we give something called monoclonal antibodies. So antibodies that can recognize a surface antigen on any given cancer. Things like rituximab, which can be used in non-Hodgkin's lymphoma, alemtuzumab in T-cell lymphoma, gemtuzumab in AML, brentuximab, Hodgkin's lymphoma. These monoclonal antibodies can be made and given almost like a drug, but they give passive immunity, meaning these monoclonal antibodies are not thought, although some people are challenging this, are not thought to lead to long-term immunologic memory. So when they work in these cancers, they work against a very specific antigen, but the concern is they may not lead to that long-term immunologic memory. They may not be able to kickstart the endogenous or the natural immune system in the way that's required to lead to that long-term immunologic memory that's so important for killing not just a few cancer cells, but killing all the cancer cells and having the memory cells in place so that if that cancer ever returned, it would be eliminated. Just like if a virus reactivates it, your immune system would come back and eradicate it if it's recognized it as foreign in the first place. However, active cancer vaccines should do this on their own. They should induce the memory, the immunologic memory you need to reprogram the immune system. And so for an active cancer vaccine to do what it needs to do, there has to be pieces of the tumor that are loaded into an antigen-presenting cell, such as a dendritic cell. And this can come in the form of peptides or proteins from the tumor, or RNA, as I just mentioned, that codes for these proteins. That can be loaded inside a dendritic cell. And these dendritic cells can then activate the T cells, the CTLs, which are CD8 T cells, and CD4 T cells. And these T cells can then traffic to the tumor and kill the tumor and kickstart the cycle of natural endogenous cancer immunotherapy. Peptide vaccines have been used and studied in the context of malignant brain tumors for children. And these peptides can be made very specifically from the proteins or peptides, the pieces of the proteins that are expressed on tumor cells. They can actually be artificially synthesized outside of the patient's body and then given in the context of an adjuvant. An adjuvant is an inflammatory material that allows the immune system to get really angry when it sees these peptides and mount an immune response. So the combination can be injected as a vaccine, and that will naturally recruit dendritic cells, these APCs, these master cells of the immune system, to pick up these pieces of tumor peptides and develop an immune response against them. These dendritic cells will then activate CTL, CD8 T cells that will then, once they're active, will traffic to the tumor wherever it is, whether it's in the brain or in the periphery, and kill those cells. However, nucleic acid vaccines are another strategy. RNA can be advantageous for a vaccine because they don't need adjuvants. RNA can act as an inflammatory adjuvant on its own. It can be, unlike DNA, that can be integrated into a person's genome. You don't want tumor DNA to be integrated in a person's genome. RNA is only expressed in the cytoplasm of cells. It can be very patient-specific, it's easy to produce and store, and can be amplified from very few cells, which is what we're doing in the context of malignant brain tumors in some of our studies. And it can be loaded into these dendritic cells, which we can obtain from patients, and it can be given in the context of chemotherapy with or without adjuvants. Doesn't need adjuvants, but you can give adjuvants to enhance the inflammatory response against the tumor antigens you're vaccinating against. And their dendritic cell vaccines have actually shown very promising survival benefits in patients with glioblastoma. These are adult patients. This is a small pilot study published by my mentor, Dwayne Mitchell, where patients who received these dendritic cell vaccines had promising, more improved progression-free and overall survival benefits compared to patients who received the unpulsed dendritic cell vaccine, the controls. However, dendritic cell vaccines still require a lot of complicated cell therapy processing. You have to obtain these cells from patients. You have to grow them up outside the patient's body. An alternative strategy would be to use something called nanoparticles, which is another strategy that I work on and others are working on, where you can just extract the information from the tumor and then just, instead of putting it in a dendritic cell, which requires many months before we actually get the dendritic cell to be active and give it back to the patient, these nanoparticles can be available immediately. So right after biopsy, we can combine this with a nanoparticle and then just give that nanoparticle as a vaccine to patients. These nanoparticle vaccines can localize naturally to patients' APCs in their body, these dendritic cells, and then these dendritic cells can activate the T cell response. So we're using both of these strategies now, where we can either take dendritic cells from patients, ex vivo manufacture these cells with RNA, educate them, give them back to patients. Again, very promising results that we've seen in patients with adult glioblastoma, but again, a process that takes many months and is very costly and complex. The nanoparticle approach could be available for everyone and bypass a lot of this complexity, because this approach can naturally localize to these cells and stimulate this T cell response. Alternatively, what folks are doing is they're using oncolytic viral therapy. So viruses can usually infect any cells, but these oncolytic viruses have been attenuated in such a way, they've been fixed in a way that allows them really to only replicate in tumor cells. The reason for this is tumor cells don't have a lot of the natural defense mechanisms our healthy cells do. And so these very weak viruses can actually replicate in just the tumor cells and then spread quickly from one tumor cell to the next tumor cell and so on and so forth, and just kill those cells. And why is this an immunotherapy? This is an immunotherapy because as this virus replicates inside just these tumor cells, what happens is your immune system then actually sees the inflammation that's happening as a result of this. And then the immune system comes in and actually ends up killing a lot more of these tumor cells than the virus does in the first place. And this oncolytic virus actually leads to a very, very strong natural immune response that kickstarts that cycle that I was showing you in earlier slides. And so some examples of open clinical trials for patients with pediatric brain tumors using these cancer vaccines, oncolytic virus. There's a couple of studies at the University of Pittsburgh, Dr. Ian Pollack, in patients with recurrent ependymomas and patients with recurrent low-grade gliomas, high-grade gliomas, and in patients with brainstem gliomas, where they're using peptides that are signals on those patients' tumors that can actually be combined in a vaccine to engender a T cell response against these tumors in these refractory settings. There's some oncolytic viral studies as well. There's a herpes simplex virus, a study, a phase 1 clinical trial at the University of Alabama in patients with supratentorial brain tumors that are recurrent. And there's a phase 1b study of attenuated poliovirus for children with recurrent malignant glioma at Duke. This is a lot of the work that Dr. Matthias Grohmeyer has been pursuing over at Duke. And so the last thing I want to talk about is targeting immunoregulation. And so there is this concept of immune checkpoints that has been shown to be really remarkable in the sense that if you could target these checkpoints, you can actually unlock a patient's T cells against their tumors. And so tumors express these molecules, these checkpoint molecules, such as programmed death ligand 1. And even if you have an activated T cell that knows to kill this tumor, it won't. It won't if the tumor expresses this death marker that kills the cell. And brain tumors do this. Brain tumors express this programmed death ligand 1 molecule. And that can turn off an activated T cell. And if you can just block that engagement, if you can block the engagement of this death ligand with the death receptor on the T cell, you can keep the T cell on and functional against the tumor cell. And so people have natural immunity against their tumors, but the natural immune cells are being basically killed off by the tumor by expression of these death markers. And these cells are basically not surviving to do what they need to do. And if you can just block this engagement, these T cells can stay on, kill the tumor, and get the cancer immunotherapy cycle going. And we've seen this in melanoma. In patients with melanoma who receive this drug called ipilimumab, which blocks a very similar checkpoint axis, we see improved survival outcomes in patients with melanoma. However, this response is only seen in tumors that have a lot of mutations. It's not seen in patients who have tumors with very few mutations. And when we think about pediatric cancer, pediatric cancer doesn't have very many mutations. Melanoma here has a lot of mutations. But when we look at something like medulloblastoma, for example, that has very few mutations. So can these checkpoint drugs actually work for pediatrics? And the jury's still out as to whether these drugs can work in pediatrics or not. But in pediatric tumors, like medulloblastoma, they may not have a lot of mutation. But what they do have is a lot of something called epigenetic modification, meaning it's not the genes or the mutations so much in these tumors that's driving them, but it's how those genes are being expressed and reprogrammed in these tumors, and specifically in a lot of pediatric tumors like medulloblastoma, there are developmental genes that should stay off that are being turned on again. And medulloblastoma can now be divided into these 4 distinct entities. And the hope is that we can harness the immune system to target these developmental antigens, because these developmental epigenetically regulated antigens should not be expressed on your normal cells. So if we can target these developmental cells, it should just kill the tumor and spare all normal tissue. There's another regulatory axis that tumors use, and it's this enzyme called IDO, indolamine 2,3 dioxygenase, which actually starves T cells of nutrients that they use to fight the cancer cell. And if you can block this enzyme that is inside these tumors, you may be able to keep the food source for these T cells intact and allow them to fight the cancer even better. And so these are some of the trials that are available for patients, the open clinical studies for patients with pediatric brain tumors that target some of these immunoregulatory approaches. One is a phase 1 study of indoximab, entemozolomide for children with progressive primary brain tumors. This is at the Children's Healthcare of Atlanta and Augusta University. Indoximab blocks that enzyme IDO that I just mentioned. And these are two studies using these checkpoints. There's a pilot study of nivolumab in pediatric patients with a lot of mutations in their cancer. These are patients that have to be screened to see if their cancers have a lot of mutations. And if they're eligible, then they'll receive this drug that blocks PD-1, that death axis I mentioned. And then there's this multi-institutional study, which we hope will answer this question as to whether these drugs can work in patients with malignant brain tumors, these checkpoint drugs. And it's looking at the safety and preliminary efficacy of this, of pembrolizumab, Keytruda, which also, this is a very similar drug to nivolumab. It also blocks PD-1, and this is for children with recurrent, progressive, or refractory high-grade gliomas. It's being run out of the National Cancer Institute, but it's multi-institutional. So these are some of the strategies that are currently being employed. Really, they can be divided into adoptive cell therapy, cancer vaccines, oncolytic viruses, or this immunoregulatory targeting. The challenges are really to induce immunity against these self, which I put in quotations, antigens. Self-antigens because your natural immune system recognizes them as self. But many of these antigens are foreign, and we can redirect the immune system to recognize them as foreign. But the challenge is to do that in a safe way without toxicity against normal cells. And to do this against what's unfortunately a very overwhelmingly immunosuppressive and immunotolerant cancer. And many of these brain tumors, like malignant gliomas in children, are very immunosuppressive. Really, moving forward, I think it will be very important to utilize combination therapies, combination immunotherapies that target these novel tumor antigens that can be recognized as foreign, in juxtaposition while we target the immunoregulatory landscape. And this may be unique to specific tumor subgroups, like specific subgroups within medulloblastoma, and it may be unique per patient. And so as we develop immunotherapy, we'll need to continually refine how personalized the approach is and really reconcile that with novel therapies that can be commercialized for patients. Certainly want to thank all the members of the University of Florida Brain Tumor Immunotherapy Program. Really, our mission is to eradicate brain cancer in adults and children. I want to thank my mentor, Dwayne Mitchell, everyone in the program. The National Institutes of Health is so important for driving research in this country. And at a time where there are proposed budget cuts to the NIH, which would deeply affect what the community can do to target diseases like children's brain cancer, it's really foundations like the American Brain Tumor Association that become all the more important for supporting our mission and getting us over the hump to the point where we can really make a difference for these patients. So I thank you. I thank you for your attention, and happy to take any questions.
Thank you, Dr. Suyor. Dr. Suyor will now take questions. If you have a question you would like to ask, please type and submit it using the question box in the webinar control panel on the right-hand side of your screen. And we do have a question here. Does immunotherapy help treat recurrences?
So I'm assuming the question is for pediatric brain tumors. So in general, yes. Immunotherapy has been shown to treat recurrences in tumors such as, melanoma, refractory cancers like prostate cancer. So yes, the data is there that immunotherapy can be used to treat recurrent cancers. The question is, can it treat recurrent cancers in pediatrics, or will it treat recurrent brain cancer in children? And that's an unknown. I think that it can. I fundamentally believe there's no reason why the immune system couldn't or shouldn't be able to be redirected against either a primary or a recurrent cancer. And the way clinical trials work is we tend to investigate these questions, especially if it's a new drug, in usually the most refractory settings or the recurrent settings. And so many of the trials I mentioned are in the context of recurrent medulloblastoma or recurrent high-grade gliomas. And really the first thing we look at is it safe or not? And that's phase 1. And then we try to answer the question, really, is it efficacious? Does it work? Does it look like it works? And that's what we call a phase 2 study. And then if it looks like it works, then we need to do the definitive study to know for sure if it works. And that's called a phase 3 study or a randomized clinical control trial, where we have a very distinct control group that gets a placebo drug and then a treatment arm before we can answer the question definitively. So I don't know whether some of these strategies I mentioned will go on to show efficacy in phase 2 studies and pass phase 3 and become a standard of care. But I think a lot of these strategies are promising based on some of the data that's in existence in phase 1 studies, and I think it's very promising, based on some of the preclinical work that's coming out regarding these things. And if it doesn't work, I do think that these studies need to be designed in such a way that we learn something so that we make sure that the next study does work and helps those patients.
Thanks, Dr. Sayor. I have another question. Are there cases in which immunotherapy is not recommended?
So there are cases where it wouldn't be ideal to use immunotherapy. And so in brain tumors, if patients are on very high doses of steroids, so steroids can be very important to manage patients with malignant brain tumors because you have an expansile mass in the brain that can cause increased intracranial pressure, swelling. Oftentimes, patients will be treated with high doses of steroids to calm that down. And I mean, sometimes you have to do that because of the risk of the tumor growing. Unfortunately, steroids are notorious for killing immune cells. And they're really, in those situations, I don't think there would be any benefit to giving immunotherapy if you're going to be killing so many of these cells or most of these cells with high doses of steroids. There are risks to immunotherapy. Certainly, even with these checkpoint medicines that I mentioned, like the PD-1 drug, we do see autoimmune diseases in those patients sometimes. I mean, again, rarely, but it happens, where even patients that have been cured of their cancer may now develop an autoimmune disease in their gut. And this can happen well after they've been off of the drug. So anytime you start to activate the immune system, and these drugs are not, they just activate all sorts of these T cells by blocking the death markers. You may see autoimmunity, and so it's something we in the field have to be very, very mindful of as we move forward.
That brings us to our next question. Besides the autoimmunity, are there any other significant side effects associated with immunotherapy treatments?
So the biggest one is autoimmunity. I think that it depends. I mean, for autoimmunity to happen, really the target is very important. So when we're talking about autoimmunity, what's the target? So in CAR T cells, the target is often a surface antigen that is expressed on your normal cells. So these genetically modified T cells, while some of the results are amazing with CAR T cells in leukemias, we see dramatic results in those contexts. But those CAR T cells target something called CD19 that's on all of your normal B cells. And so in leukemia, they lose all of those cells. And these patients can't make antibodies anymore and need to be on IVIG for life, which is very manageable and better than having cancer. But is a side effect of that type of a therapy, that CAR T cell, because those T cells are targeting a normal antigen that happens to be on all of the cancer cells. And so depending on the platform and the target you're going after, really determines what kind of autoimmune effects you may see. With the total tumor RNA strategy, when we just extract the RNA from the tumor, we're allowing the natural immune system to decide which of those RNA antigens inside the tumor are foreign. And in that strategy, we haven't really seen autoimmunity in either our preclinical studies in mice or in our human studies in patients. There was one patient who got dendritic cell vaccines, an adult patient with glioblastoma who developed a side effect, an autoimmune reaction essentially from the dendritic cell vaccine. But we did the test and it wasn't the dendritic cell vaccine, it was actually the GM-CSF, the adjuvant that was with the vaccine. This patient developed a reaction to that. So in that strategy, we really haven't seen autoimmunity. So really, when we talk about autoimmunity, it depends on the context. CAR T cells, definitely. We can see things like cytokine storm, where patients can get flu-like illnesses and get very sick very fast with that medicine. Things like dendritic cells, less so. We haven't typically seen autoimmunity in that context. Again, those drugs, these new drugs like Keytruda, nivolumab, you can see very distinct autoimmune syndromes, well after discontinuing that therapy. So that's really the big one, is autoimmunity. Certainly, anytime you give an immunotherapy product, you may incite the immune system in such a way that you get, you may experience a flu-like illness. So, fevers, chills potentially, myalgia possibly. All this could be from the recruitment of the immune system itself.
Thank you. I have another question. Are teens with adult-type tumors more likely to benefit from immunotherapy since they have more mutations?
So, great question. Yes, yes and no. I think that for the checkpoint inhibitors like PD-1, the data seems to suggest that patients who have more mutations have better benefit from these drugs. However, what's unclear to me are we only seeing a benefit in those patients with more mutations because they already have a pre-existing immune response? Or is it really just because they have more mutations? So when you have more mutations, it may be more likely that you have a better or stronger endogenous immune response. And that's really what these checkpoint drugs are probably doing. They're just rescuing an endogenous immune response. So let's bring it back to pediatrics. If pediatric tumors don't have a lot of mutations, they may not have a very strong endogenous immune response. But that doesn't mean we couldn't generate it. That doesn't mean we couldn't vaccinate those patients and generate that endogenous immune response, and then come back with these drugs like PD-1 and rescue it and allow these cells to divide and proliferate and do all the things they need to do. So it's a good question. It would seem that more mutations allow these drugs to work, but I don't know that you need to have them to have checkpoints do what they do, or many of these drugs do what they do. In the sense that if you can just develop an immune response, you may be able to have that response synergize with an immune checkpoint like PD-1 drug. And I don't think mutations are the only thing that needs to be targeted. I think in pediatric tumors, developmental antigens, these things that are supposed to be expressed when you're a fetus, that are not supposed to be expressed in in a normal child, but that are re-expressed in tumors, those are perfect antigens to go after and develop an immune response against, because it shouldn't cause autoimmunity, presumably. And so I believe that the immune system can be sensitized against anything if you do it with the proper kinetics, using the proper formulation, I certainly, there's no reason for me to think that we shouldn't be able to do this against developmental antigens or other type of antigens in pediatric tumors if they don't have a high burden of mutations.
Thanks, Dr. Sayur. That is all the time we have for today. Thank you all for joining us, and thanks once again to Dr. Sayur for his wonderful webinar presentation. Besides our free educational webinars, ABTA has a variety of programs available to help connect patients and caregivers with information and resources to help support them in their brain tumor journey, as well as publications and resources for healthcare professionals. For more information, visit ABTA's website at www.abta.org or call the ABTA Care Line, which is staffed by caring professionals, at 2282. Let's pause for just a moment to conclude our webinar recording.